In this video, we will understand these M1-type macrophages are augmented by the activated CD8 plus T cells, and also these macrophages use phagocytosis to kill the tumor cells. So we will understand how they come up to this point. So in order to check whether the CD8 plus T cells augment these M1-type macrophages, they designed an experiment that is shown in this panel A. So what they did is they took the bone marrow from the mice, and in one case they cultured them with their supernatant, and then use this IFN gamma inhibitors to check whether they have this the macrophages or not. And in the other case, what they did is they isolate the CD8 plus T cells and then use those activated CD8 plus T cells supernatant in the bone marrow of the same mice models, and then perform macrophage stimulation in CD8 plus cells supernatant. So this is what I just said, and in both the cases then they analyze the cells. So in the one case what they did is they cultured them in the same supernatant, and in the other case they cultured them in the CD8 plus activated CD8 plus cells cultured medium. So it has the CD3 CD28 peptides, whereas in the first case it only has the bone marrow supernatant. And thereafter, after 1 day, after 0 day, they analyze this MCSF, and after 1 day they also add this cytokines, that is IFN gamma and LPS, to check whether it will further augment the M1 macrophages or not. And what they have find out is that percentage of the macrophages are higher where in the second case, where they use the supernatant from the CD8 plus cells isolated from the mice. And for the unstimulated unstimulated bone marrow conditions is shown here. That is, in that case they do not use the cytokines such as LPS and IFN gamma. And when they use LPS and IFN gamma, they see a slightly more increase compared to the unstimulated BDM. But in both the cases, the signals were higher where they used supernatant or cultured medium from the activated CD8 plus T cells. And thereafter, they wanted to check the different factors or different gene markers that are present in this sample. So what they check the INOS, CSCL, MSC2, CD86, and other cytokines. They check at different conditions. That is, first one they use the direct medium, and the second conditions they use LPS. Third condition they use B16F10 plus bispecific antibody supernatant, and in the other case they use B16F10 plus bispecific antibodies plus CD18 supernatant. In the last case they add all the things, that is B16F10, bispecific antibodies, CD18 supernatant, and LPS. So here they add all the things, and what they find out for the INOS, they see a significant increase in the last conditions, and this increase continues for CXCL9, but it goes down for the MSC2. And when it comes to this other markers such as CD11C, CD163, in this case also we can see the increase is highest when they use bispecific antibodies and CD8 plus supernatant and LPS. So together they shows that in case of either cytokines or the CD8 plus supernatant, there is an increase in the percentage of macrophages, indicates that the activated CD8 plus T cells augment the macrophages. And now using the Wishbone trajectory, what they find out is that there is an increase in the macrophage occurs early in the stage, whereas increase in the INOS occurs later in the stage. So this indicates that this activated CD8 plus cells cause early augmentation of the M1 type macrophages. and now they wanted to check whether the M1-type macrophages is increases or M2-type macrophages also increases. So they use the different markers for the M1 and M2-type macrophages, and what they have find out is that when they use the activated CD8 plus T cells supernatant along with in the tumor lysate, they see a increase in the markers for the M1-type macrophages such as ONS2, CXCL9, but when they check for M2-type macrophages, they see a decrease in the M2-type macrophages such as this HMOX1, CD163 for this tumor lysate and CD for the conditions of tumor lysate and CD23 and CD28 supernatant. So this indicates that M2-type macrophages does not increase with the activated CD8 plus T cells, whereas M1-type macrophages increase. Next, they wanted to check the percentage of iNOS macrophages since they are the late augmented populations. They wanted to check its percentage at different conditions. Or one observations, yeah. Before we go to the results of the final, this panel F. Why they did this experiment, we need to understand that the results that they get from the in vitro experiments are not completely in line with the results that they get from in vivo experiments that are shown in the figure 2. You can refer to the previous videos. So the hypothesis that they made is that the toll-like receptors could be present on the tumor cells that might also get activated in the in vivo studies. So they did the experiment with different inhibitor of the toll-like receptors, and how they select the inhibitors, they use the transcriptome analysis of the tumors and find out that the tumor TLR4, TLR79, TLR12 are present in the tumor cells. So they use different inhibitors, and what they have find out is that the percentage of iNOS macrophages goes down in the case where they use this TLR4 inhibitors, and also it goes down when they use this anti-IFN gamma or anti-IFN GR inhibitors. So indicates that TLR4 and IFN gamma and IFN GR plays an important role towards augmenting the iNOS macrophages. And these things they have validated, and after validating this thing, they wanted to check whether the cytokines based tumor killing occurs by the macrophage or there are some other tumor killing occurs. And from the transcriptome data, they find out that the markers for the phagocytosis are higher. So they wanted to check whether the phagocytosis cause the tumor killing or not. So they check the phagocytosis in different conditions. First one is the unstimulated RMA QA1 cells, and in that case they find out is that when they use activated CD8 plus T cells, they see the highest when they use this GHG supplement, this thing, the supernatant from the CD8 isolated from the mice, they see highest phagocytosis markers, and this phagocytosis markers is highest when they for the conditions when they use the stimulated supernatant that has this LPS and IFN gamma. And same things they have find out for the B16F10 mouse model condition also. That is when they use the CD8 plus CD8 plus activated CD8 plus cells supernatant, they see a high increase in the phagocytosis. But this increase is becomes further when they use the activated or when they use the stimulated supernatant, that contain LPS and IFN gamma. So all this together indicates that the CD8 plus T cells not only augment the M1-type macrophages, but these M1-type macrophages use phagocytosis to kill tumor. I hope you understood this conclusion, how they come out to this conclusion. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.